Today we're going to talk about reconstructing metabolic networks in prokaryotes. We're going to follow the E. coli lecture we just went through last time, but look at these models in much less detail than we did for E. coli. Many of them, of course, I would say most of them, of course, are much less well developed than the E. coli model. So first, let's talk a little bit about which reconstructions are available, which organisms are covered in the literature so far. Then I want to talk a little bit about pathogens and infectious disease, because there are a lot of opportunities now to move into detailed study of infectious disease. Not only the common pathogens by themselves, but also variations of them, multi-strain type reconstruction projects like I mentioned for E. coli last time. There are some reconstructions now available for photosynthesis in cyanobacteria. We'll talk a little bit about those. I'll mention the activities that have been focused on microbial communities. And I think all of you know that this is a topic of exploding interest, really, not just from an ecological standpoint, but also from a human health standpoint. As I think everybody has been sensitized to now, we have a lot of ecosystems living on us and in us, and they interact with the human body quite tightly and are integral to wellness and disease. Then at the end, we'll talk about a bacteria called Geobacter, which is a little bit of an unusual soil bacteria. But there is now a 12-year history of studying this organism in detail using genome scale models. It was poorly known when that process started, but genome scale models really helped to drive discovery processes around this organism and have helped develop a more detailed understanding of its physiological function. So that's a good kind of case study to look at. All right, so talk a little bit about the available reconstructions and what they've been used for. We get a little bit of a taste of this in the last two lectures. So we'll say a few more things about them. So first, the phylogenetic coverage of models is shown in this diagram to the left. Most of the reconstructions available right now are for bacteria. Enterobacteria, of course, as a class are well known, and so there are many good enterobacterial reconstructions. Don't know what to call that color there, but the kind of light red color shows the archaea that have been reconstructed, so there are some of those available, and the light blue branches on that tree are the eukaryotes that have been reconstructed and analyzed. And the bar chart next to the phylogenetic tree shows the progress in terms of published reconstructions, and there are about 100 of them now. At the bottom is also an interesting historical profile. It shows the number of unique reactions that have been placed in reconstructions now, and the total number of those is now between 14 to 15,000, as I recall. And that little bar chart to the side shows what coverage of EC numbers is represented in these reconstructions, and that's about 60%. So there are still 40% of EC numbers that corresponds to the reactions that are not found in any reconstruction yet. So progress is being made, but there is some way to go. And I reviewed this with you last time. When you look at these reconstructions and try to figure out how similar they are in terms of their reaction content, it turns out that they are quite similar, most of them. They are shown in that green swath in that PCA plot. And that's a reflection of the fact that most of them contain the basic metabolic pathways in them, but not those that are particular to a species or an organism. For a few organisms that's happened, as the panel to the left shows. The photosynthetic organisms, of course, are quite different than the rest and are shown in that light red area. And then the enterobacterial reconstructions in yellow, getting more and more specific to enterobacteria. And then the blue one, that blue segment that's stretching out from the origin of that plot represents the successive reconstructions of yeast. So those are becoming more and more specific and different than the rest. Now, an assessment of these reconstructions in detail shows that there are some challenges ahead. One is to help develop experimental methods that actually help to elucidate the reactome. We have good ways to sequence now to get the genomes. We have good ways to deal with transcriptomes. We have good ways to deal with proteomes. Metabolomic profiling is pretty standard now. But we can't really figure out the reactome easily by any high throughput method, but some of those are being developed. The second side of this pentagon calls out the fact that the hard work associated with curating the bibliome associated with a particular target organism, that work needs to be done. And it hasn't been done well for many of these target species, but that manual labor that goes into that just, needs to happen so we can get more high quality curated models done. And the bottom side of that pentagon calls for an increased phylogenetic coverage. If you look at the tree that I had before, it's very much focused on enterobacteria, with just a few other phyla being represented. And as I recall, there are 27 major trees, branches on the phylogenetic tree, and only a part of them have any reconstruction in them. So that needs to be done better. Okay. There is a review out and available from now a few years ago that reviews the uses of these reconstructions of all organisms. And the use classes are very similar to that for E. coli. There are metabolic uses, there are discovery uses, there are in silico network characterization uses, and there are also uses now for community, microbial community applications that I'll mention here in a moment. So similar sorts of applications as for E. coli. And as I mentioned at the beginning, we'll talk about Geobacter as a specific example here in a moment and show some of the detailed results achieved based on the Geobacter reconstruction. Let me talk a little bit about infectious disease and human pathogens. I believe that this is an area that is very ripe for rapid progress for this type of systems biology. There are many, many microbes that live on human beings and in human beings. Most of them are commensal. They live in harmony with us, but a subset of them are pathogenic. And if they go out of control, bad things happen. So this diagram here shows the different reconstructions that have been performed for human pathogens. So in the top right corner, the two pathogens shown in there are Staph aureus and Pseudomonas aeruginosa that, along with E. coli, form most of the infections treated in hospitals in the United States. I think these three pathogens account for almost 90% of the cases treated. There is a good reconstruction, of course, for E. coli. There is a great reconstruction coming out for Pseudomonas putida that probably needs to be adapted to aeruginosa. These two organisms are somewhat related. And there are good reconstructions for Staph. Staph metabolism is not as diverse as that of E. coli and Pseudomonas. Then there are a number of other pathogens shown on there. Further down in that column to the right is Helicobacter pylori, which I think is of special interest because it's a very simple genome. It's on the order of 1.7 megabases. It's a simple organism, highly tailored to living in the stomach, low pH, so forth. It affects every other person on the planet, mostly non-clinical infections. People may be infected by H. pylori and not know about it for many, many years. So it keeps irritating the lining of the stomach and is a causative agent in stomach cancer. And there are many, many genomes that have been sequenced of Helicobacter, and some detailed studies of its transcriptome and its regulation have been carried out. There are very few transcription factors identified on that genome, so it should be a relatively simple organism to get a good model for. And I think I mentioned the same opportunity exists for Thermotoga, an environmental organism, and a thermophile. So most of these are bacteria, but there are a few eukaryotes on that list. So I think that over the next decade, reconstructions for these organisms will progress and develop towards understanding infectious disease very well, and possibly lead to new and more effective treatment modalities. So that statement is based on a collection of initial studies that have been done with these organisms. And there is a review article on that that's now 2 years old that you can't read, but let me explain to you what's in that table. The rows in it are the different pathogens and the disease that they cause. These models have been studied with a suite of different COBRA methods, and we'll talk about them later. And they have been used to characterize their physiology, suggest drug intervention, and so forth. Some of these have entered an experimental phase, and if you look down these three last columns and look at the X's, you can see where validation has been done on these models, either with retrospective data from the literature or using new data. And for some of them, if you look at the last columns, there are some check marks in them where drug discovery and drug intervention has actually been characterized and looked at. So I think it's easy to see that a table like this will fill in quite nicely, I think, over the next decade. And there, well, I would say that this table represents a target-rich environment. There are a lot of exciting things coming up with systems biology of infectious disease. And we're talking about bacteria now, mostly. Potentially some of these eukaryotic organisms, but not viruses. They have not been dealt with yet. So that's a very interesting area of study. Photosynthetic organisms, you know, a large fraction of the metabolic activity on the planet is photosynthesis and carbon fixation. And these are mostly done by unicellular organisms in the ocean, either prokaryotic organisms, cyanobacteria, as I mentioned, or unicellular eukaryotes that we'll talk about in the next lecture, and then, of course, plants on land. Synechocystis is one of the model organisms for bacterial photosynthetic organism. It's genetically manipulable. It's a simple organism. A lot of data available on it. And there have been many successive reconstructions of this organism, as indicated in that table. And a couple of years ago, they were all put together into a consensus reconstruction, and that reconstruction was expanded and curated by Juan Nogales, who spent a long time on the manual curation and came up with a very well validated models. And it shows a few things summarized in these 3 bullet points. There are 2 main states of its metabolism, as you might expect. There is the photosynthetic apparatus under light conditions, and then the CO2 limited state during fixation. There were and as I'll show on the next slide, detailed analysis of the pathways that are involved in photosynthesis and carbon fixation. And these are represent certain trade-offs. And so there's a notion of organism optimization that came out of this. And there is an inherent robustness issue in that trade-off that is also studied in that paper. So very well curated model, validated against a lot of data, been quite insightful in terms of understanding the physiological functions of this organism and the fundamental trade-offs that are involved in its lifestyle. So every species may make those trade-offs in a slightly different way, depending on their evolutionary history, maybe geographical location, and so forth. So this is just a snapshot of the photosynthetic apparatus. I'm showing this because for this organism, detailed reconstruction was achieved. And this is one of those membrane processes that I mentioned earlier that proves to be so hard to reconstruct. But for specific cases, detailed reconstruction is achievable, and this has been done for Synechocystis. And based on this, there is a number of resource groups now interested in making a collection of different reconstructions of different variations of Synechocystis. And I think I also mentioned last time, and maybe should do it again, that when we build a model for a strain, it becomes a strain-specific model. But if it maps on to many strains in that species, it starts to help us understand what is a species and what may be the basis for strain variation within a species. And this is a somewhat new angle in the use of metabolic reconstructions, and it's enabled by the fact that we can sequence now so rapidly and so cheaply. Okay, so those are some comments on bacterial or prokaryotic photosynthesis. Let's talk a little bit about microbial communities. There has been a lot of interest in studying them. And again, this interest comes from progress in sequencing. And when the cost of sequencing started dropping so rapidly around 2007, a lot of money was spent on sequencing microbiomes. And of course, there's a special interest in the human colon, because the species composition in the human colon has strong correlates with human disease and human wellness. Now, this represents an interesting challenge. So here is a diagram on the left that I've shown before, this kind of modularization of the genotype-phenotype relationship for a single strain, where we go from the sequence, identify the molecules, build up the modules, end up with a genome scale model that allows us to predict the physiological functions of this organism, and typically in isolation, studied in isolation. If you draw an analogy with communities, there is a process that needs to unfold here, and this should unmask. Yes. In a community, we have many members, like we have many protein molecules that make up a species. Now we need to characterize them individually. And if we think we have a good model of individual species, we might be able to model and study interactions between a small number of bacterial species. Scaling that up to hundreds, if not thousands of species that may be living in the human gut is a process yet to be defined. We don't know if that's possible or not. But at the community level, though, we can do certain top-down studies by characterizing composition of the community. You can look at perturbations to it and see how that species composition changes. In the pink box there, I should say that there are now efforts that have been undertaken to look at how two species interacts, two strains interacts of a species, or two species interact. And Karsten Zengler here at UCSD has driven an effort to characterize a 5-species interaction and how that takes place under multiple different conditions. So this is perhaps a grand challenge, is to flesh out the right-hand side of that diagram. So progress is being made from bottom up, progress is being made from top down, but those two haven't merged yet, but people are beginning to try. So there is some set of applications of community functions that have been studied, and they are summarized on this diagram. If you look to the top, there are a few studies that have been made of mutual auxotrophies. So you have two species that need the nutrients, typically an amino acid. They can get it from the partner of the community, so they exchange amino acids. And they very rapidly learn how to do this when they try to evolve interactions between two mutually auxotrophic species. So that's kind of interesting to study these, predict them, maybe make synthetic auxotrophies like this, and try to learn about how two species interact. We go in a clockwise fashion here. In the, I guess, 3 o'clock position, there's a graph that shows the composition of a community of two species, Rhodophyrax and Geobacter. And you can build a model of how they exchange nutrients, electron acceptors, electron donors, and so forth. And you can actually calculate the optimal composition of that community based on the environmental conditions. And those predictions, remarkably, in the few studies that have been carried out, prove to be accurate. You can predict the optimal metabolic subtasking in a community. If you look to the bottom, the 6 p.m. position on this diagram, you see a macrophage and TB. So you can actually study now tuberculosis in a macrophage individually as isolated organisms, but you can also put it together. So you can simulate an infected macrophage. And it turns out now that you can get the expression profiles in the unaffected state, the separated state, and the infected state. You can pick out the transcripts somehow from the pathogen and map them, I guess, onto that genome and separate them from the transcripts of the host. And to the bottom, you see some histograms that are in blue and kind of a reddish purple color. And these are histograms based on random sampling of all the candidate flux states of the macrophage, unaffected and infected. And you can, based on this and the data mapping, infer what the shifts are in the pathway usage in the host after infection. And interestingly enough, what seems to happen after a TB infection is that it promotes a suppressive metabolic state in the macrophage, so it doesn't activate as well. So we'll talk maybe more about this later in the class. Then if you go around to the 9 o'clock position on this diagram, there is something there that shows how gene content is predicted to change in an organism over long-term evolution, sort of speciation and simplification. And it's actually remarkable that the temporal order of such events can be predicted. So this is probably just an initial set of applications of bottom-up systems biology models of metabolism to communities, their behaviors, and their properties. Okay, so those are general comments, and like I said, we may talk more about them later in the class. But let's finish this lecture up by looking at a specific case. And that is the case of Geobacter sulfuriducens. And this story started soon after the E. coli model came out first in 2000. I seem to remember 2002, 2003. And there was kind of there's been almost a 10-year history of studying the systems biology of metabolism in this organism. And this was enabled by a rather generous grants or funding from the Department of Energy. And you see in a moment why they are interested. And this was largely driven by a prominent microbiologist by the name of Derek Lovelace at the University of Massachusetts that knows the ecological function and behavior of this organism very well, in addition to being able to study its genetics and biochemistry. And he worked with systems biologists that built the reconstruction and helped to use it to design experiments, interpret data, and so forth. So this is an organism colored in green in this image shown over there, and was colored in pink in the background are iron particles. So it turns out, as I'll talk more about in a minute, that this organism has very unusual ability to use electron acceptors. And it can actually make iron accept electrons and reduce iron. And there it is growing on an iron clump. It has nanowires. If some of you have been reading microbiology in the last few years, that can be hundreds of microns long, but I forget not the width is a few tenths of nanometers, and they are conductive. Apparently, they can conduct electricity, and so this is one way apparently in which this organism can just latch onto iron ore and induce this electron exchange. Okay, so what is the key feature of this family of species? They are able to reduce a lot of metals that live in the soil. And the fact that they can dump electrons onto uranium made the Department of Energy very interested in this organism, because of uranium contamination in groundwater. Because if you would promote geobacter growth, say, in the ground, they might fix the uranium by reducing it and stopping it, and it wouldn't be flowing as an ion in the groundwater. So it's not only uranium, but there are other heavy metals apparently it's capable of reducing in this manner. So I think Derek discovered this organism in mud in the Potomac River, I think in the late 80s or early 90s, I forget now. I discovered this, this whole family of organisms. They are what you might call bottom of the barrel organisms, because they live on the worst substrates. They live on acetate and have a specialized lifestyle to live on acetate. And if they live in a community with other organisms that may take some more complex carbohydrate source and secrete the acetate, a geobacter will take the acetate and use it. And that's kind of a really poor quality substrate, but usable. Needless to say, therefore, a lot of the discovery of metabolic capabilities here was focused on specialization to use acetate as a substrate. So it's not just import and assimilation of acetate, but also it was discovered that geobacter has more efficient routes, metabolic routes, from acetate or acetyl-CoA to amino acids than you find in other organisms, which makes perfect sense based on the physiological niche or functions of this organism. So the reference at the bottom of this slide is to a paper in Nature Reviews in Microbiology that details this history. So just show some of the highlights. So the transporters to the left of that top diagram show that there is a plethora of acetate importers encoded on this genome with different Km values. So different ones are expressed depending on what the concentration of the acetate is in the environment. They have transporters with really low Km values, so they can scavenge acetate from the environment. So this was detailed. Then, once acetate is inside the cell, you see a blue, green, and a black box. It has a number of different metabolic processes to activate acetate, which is to convert it into acetyl-CoA, which, as we know, is a basic biosynthetic precursors in a cell. And so that was elucidated. And what you see to the right of the top diagram are these nanowires hanging out of the cell, loaded with cytochromes. And you see that kind of purplish box at the bottom there. There was a discovery of some of the components in that electron transfer chain that takes the electrons off the metabolites and put this into that nanowire, and where it's ultimately then delivered to the metal that receives them. So these metabolic processes were enabled by using the discovery of these processes was helped, aided by, and in some cases one would say enabled by the metabolic models. An example of that is shown at the bottom here, that acetyl-CoA goes to, I think it's valine or isovaline in this case, through a different route in Geobacter than it would in E. coli. And when you calculate the energetics of that process and the efficiency of that process, you see that this is a superior pathway in terms of yields and energy use to the routes that Enterobacteria use. So this is a reflection of the fact that this organism is more specialized in its metabolism to live on acetate. So even if these reaction substances are known here and there, the way a particular organism uses it may be highly customized. And this is what we mean by a functional state. And we are very interested in finding the optimal functional states of metabolic networks that support a particular lifestyle. And this is something we will get into in detail in the third part of the class. I thought I should show you this too. We talked about multi-scale models going from a genotype to a phenotype. Well, there are many other multi-scale models. What is shown to the top left here is a geological model. So there are geological models that predict the flow of water underground as a function of porosity of the rock and so forth. And you can now place in those models, as shown in that little call-out box there, Geobacter. So this Geobacter is actually in the ground at a certain density, certain species. You can add into that simulator how effectively they would fix these ions that would be flowing in the groundwater to predict the fixation rate. And so there's some data on that shown in this diagram. So people go out and do field studies and try to study the coupling of the function of the organism with these geological models. Okay. So that's a brief survey of other metabolic models available for prokaryotes than E. coli and the next of kin Enterobacteria, where the models have reached a high level of sophistication. So there are many metabolic models available for bacteria in other phyla. They are not of the same quality as the enterobacterial models, as I mentioned, but there is no reason why they couldn't be reconstructed in more detail, because the methods, the workflows are now available. So if such projects are financed, you could be able to develop these models to a much higher degree of sophistication and address these applications that I've been mentioning throughout this lecture with much more certainty. So their quality and scope varies as outlined in that pentagon that I showed you. So the issues are now defined in terms of what you would need to deal with if you wanted to improve their quality. So some of the applications that I mentioned in here are infectious disease, and I think this will grow rapidly in terms of application and practical use over the next decade. There's the photosynthetic application. It turns out that a lot of the genes involved in photosynthesis in lesser characterized organisms are unknown. So one of the challenges associated with this reconstruction is simply to find the functional genes. So this is at an earlier stage of development than, say, the application to infectious disease. And then the third bullet point there is microbial communities. A lot of interest in developing this capability, some initial successes, but a long and exciting road ahead. And I showed you what does happen if you get financed to study an organism that is poorly characterized by talking about the history of the Geobacter reconstruction. That is really a success case and shows how systems biology can drive forward experimentation and the understanding of a new bacterial species. And in particular for Geobacter, it has been engineered also for some, what should I say, unusual metabolic properties. So that's a summary of reconstructions of prokaryotic metabolism. So thank you for your attention.